I will talk a bit about the general idea of brain observatory, and will then do a quick dive into one of them, the neural pixel one, and talk about the open scope. And then Dr. Saskia de Vries, who's an associate investigator at the Allen Institute, in the second lecture, she'll talk in more depth about one of the observatories. In particular, she'll tell you about the tutorials. She'll show you how to access the data using SDK, because there's untold amount of data there that could be mined, for instance, for projects, in case some of you are thinking about projects. And then tomorrow, totally separate, I'll talk about the neural basis of consciousness. So the vision of this brain observatory about 10 years ago, this is together with Clay Reed, who joined us from Harvard at the Allen Institute, was to build something similar to what's common in astronomy, i.e. where you have, well, we can record cellular level brain activity because the atoms of perception, of memory, of intelligence and consciousness are neurons and their ensembles. So we need to be able to record at that level of granularity, to do it brain-wide, to do it in a very routine, in a very reliable, and in a high throughput manner. So reliable, in other words, with high statistical reliability, and in a way where you could do day in, day out this observation, rather than one-off experiments that take you many years to set up. This would be run just like a astronomical observatory by professional staff, rather than by graduate students or postdoc who work on one particular project for their PhD or their post-PhD work. And several sets of instruments. So we are talking about 2-photon and 3-photon, single plane and multiplane fluorescent imaging. I'll talk a bit about that, but mainly Saskia, and then high-density electrical recordings. So the two complementary techniques that have different windows onto the brain. And then finally, to open this up to the community at large, not just for internal experiments, but also for people like you or anyone else on the planet face. And we've just done this and released a request for proposal. So I'll talk about that at the end. So all of this in biology, just like for real estate, location is everything. So it's very important to know where you're recording from. It's not just, you need to know not only this is a neuron, let's say in primary visual cortex, but where in primary visual cortex, since fovea is very different from the periphery. So already many years ago, we built an increasing set of more finely grained atlases. The last one is called the Common Coordinate Framework version 3. It's used now throughout the field in the mouse. Where we have because we can breed many mice, and we can use transgenic animals. We can do it in there, for example, you see in the top image is an image of 1600, the brains of 1600 mice morphed and superimposed onto each other. So essentially, it's a high resolution atlas with 500 million pixels with a resolution of 10 micron. And everything we do, so all the cells that you can access, for example, on our database, you get a coordinate system, X, Y, Z, and you get an area assignment. Where is it in what layer or what particular region of the cortex or in subcortical structures. And so everything we do, is it electrophysiology, transcription, etc., carries this spatial tag. So we built this pipeline. So by pipeline, we mean a set of common procedures that we do over and over again, where in this case, in one end, sort of enter mice, either wild-type mice or particular mice that express particular, genes that we've introduced. And then over, weeks or sometimes months, they progress very systematically. We have animal husbandry, we do standard surgery, we do intrinsic imaging that's akin to fMRI, where we localize, in our case, because of much of what we do is around vision and visual perception. We sort of localize the various visual areas, lower, V1 and higher order visual areas. The animal goes through standard behavior. And then we do the physiology in behaving animals. So these are all head-fixed animals that imposes some constraints. These are not freely moving animals. And the physiology can either be electrophysiology or optical physiology. Then after some time, in this loop between behavior and physiology may go on for some time, right? Because in some cases we train the animal behaves in multiple sessions, let's say up to 10 sessions. And then the animal gets sacrificed. We reconstruct the brain with high resolution tomography. So we process that, we process all the data, and then we spend a lot of time in this last step, the data processing, and to move all the data onto the web, into the cloud. So the data has to come in a standard format. We use a format called NWB that we help to develop with the community. And as I said, we have to identify where the neurons are located and assign tags to the activity of each neuron, plus all the metadata about the animal, what was done to it, its training history, etc. So it's a very elaborate set of methods. And then we obsess a lot about quality assurance and quality control. In other words, we try to build devices, tools, reticles that are very precise, so we can, let's say, return with high precision to the same place in the brain, that the surgery is always the same. And then after we acquired the data, we do a lot of quality control where we throw out quite a bit of data if it doesn't, if it falls short of the QC criteria. So here we have, all sorts of machines and widgets. We have a significant engineering department that helps us build all these tools and run and execute them. So here you can see one movie, a mouse watching, in this case, a natural movie, the movie Touch of Evil. And so you can observe it here on the left. Running on the right, we track, for instance, its eye movement, so its pupil side that tells us something about arousal. We also have other cameras that track its body, its position of its tail, etc. And here you can see in this particular case, you can see in live the activity in a plane of excitatory layer 2-3 neurons. This flickering light is, essentially the correlate of the mental activity of the mouse, what the mouse is thinking or deciding or seeing as it runs past these various visual stimuli. We've done this. This was our first database data set that we released several years ago, more than 60,000 cells. And Saskia, as I said, will speak more about this and related data sets. So we also helped develop, together with the consortium of the Wellcome and Howard Hughes Medical Institute, particularly Tim Harris at Janelia, we helped to develop sort of what's now state-of-the-art technology, which is neural pixels. So neural pixel, think of you have a long shank, 10 millimeter for the mouse. Now for the non-human primate, it's 49 millimeter long. So it's a stick of silicon, highly machined, and there are roughly 1,000 electrodes along this stick, and we can select 384 of them. They're spaced in the standard version, they're spaced 20 micrometers apart. Here, the central image is a high resolution computer model of our model of primary visual cortex. So this is in the mouse, it's a little bit less than a millimeter from top to bottom. The top is the top of layer 1, and the bottom is the bottom of layer 6. And you can see sort of the individual raised electrodes on that piece of silicon, which is 70 micron by 22 micron thick and wide, and as I said, 10 millimeter long. It's a fantastic technology because all the amplifiers, multiplexer, digitizer are in the base. It has very high signal to noise. And so you roughly have 50, so per millimeter cortex, which is what many people are interested in, you've got about 50 electrodes. And so this allows you, because you can pick up neurons, typically one neuron you can pick up from many electrodes, you can do really nice what's called a spike sorting, where you try to identify you can listen, out of all the chattering that you hear, you can sort of listen which neuron assign an identity to them. So typically, we record several hundred neurons, and in our pipeline, we have up to 6. We typically have 6 of these neural pixels inserted into an animal, and so we can now routinely record 1,000 neurons on individual trials. This technology is fantastic, and there are already successor technologies. So there's Neural Pixel 2.0 was just published by Steinmetz et al. In Science, where essentially it's the same thing, but now you have 4 shafts. It's like a comb. You have sort of four shafts parallel to each other, still limited 384 electrode. There's Neural Pixel Ultra that we're playing around with, where the pixel density is an amazing 4 micron. So you can really see sort of even tiny neurons, interneurons, that you don't pick up with a standard pixel, and that you don't pick up, of course, with any other electrode. You can do this. There are so-called Neural Pixel Opto that have been developed, where you have light-emitting diodes of 2 different lights, so you can drive with optogenetics. You can not only record electrical, but you can also drive optically. There's a Neural Pixel for a non-human primate version, and of course the first Neural Pixel human experiment has been done as part of human neurosurgery at MGH. And they are further developing Neural Pixel 3.0, which uses much smaller technology, going down to 65 nanometers or even 30 nanometers, and so packing even more electrodes onto the stick. So this technology now is very mature, where we estimate we can probably pick up, I don't know, some significant fraction of all the neurons within the listening radius of the electrode. All right, so using these 2 technologies, 2-photon or 2-photon single plane imaging, neural pixel recording, and 2-photon multiplane imaging, where you can simultaneously image in 2 different places, let's say V1 in a higher order visual area in different planes. We've now released this data, and you can find this. And as I said, Saskia will talk more about it, and the SDK and processing some of the data. We've released sort of on the order of 200,000 neurons involved in many sessions in animals running or behaving. Most of that data has never been particularly deeply analyzed, because there's so many questions you can ask, and we have, of course, limited manpower. But other people are using this. I'll show one slide. Yeah, in fact, since we published this data, so for example, the visual coding public data set, the first data set we published, there have been just on that optical physiology database more than 12 external publications. So people who've downloaded, people like you, sometimes at summer courses, who've downloaded some aspect of this data, all of the data, and then did various discoveries, did deep mining and discoveries in there, and published their paper. So this is really moving towards this model that's in astronomy, where you have these large scale, let's say, Sloan Digital Sky Survey, where one group will do this extensive survey of the sky in a particular wavelength, and then make the data available to everyone, to the entire community. And then, different people in that community will do their own data mining and their own discoveries. The advantages is this, because all the data is there, all the metadata is there. We have hundreds of pages of protocols we publish, where we detail in great detail what we do. So it leads to much more reproducible science, and of course, open science. So, which is part of the ethos of the Allen Institute, big science, team science, and open science from day one. All right, so with the neural pixels, we are now recording so typically 6 neural pixel electrodes in one experiment. And so we can pierce here on the left, you can see that we can record from not only primary visual cortex, but also these higher order visual cortices. Think of them like the V2 and V3 and MTs. And in the primate, the mouse has fewer visual areas, maybe about 12. And we can record from them because they go through the entire, these electrodes are 10 millimeter long. That means the brain of a mouse comfortably fits into a sugar cube, so it's under a cubic centimeter, compared, let's say, to a human brain that's more like a liter, 10 by 10 by 10 centimeter. It's roughly 1,000 times larger, the human brain compared to the mouse brain. The surface area from cortex is 1,000 times larger, and the number of neurons, it's like in the in cortex of a mouse, it's roughly 14 million. In the cortex of a human, it's roughly 16 billion, but they are quite similar, of course. So here we can record from cortex and from thalamus at the same time. In these, while the animal, you can see on the right, it's running on this disk. So these mice love to run. So they run on the disk while we show images to them, or movies, or they're trained to detect particular images or changes of images while we record from their brain. We can do this since, as you probably all know, there are no pain receptors in the brain proper. So with proper surgery and no infection, you can do this without the animal experience any distress. And so here you can see, for instance, this is a paper we published. I'll be speaking now more about it. We published a couple of months ago, based on these particular experiments, where we can record the stimulus. We record from these different brain areas. So the first 6 are cortical areas. The next 2 is LGN, so that's the relay station between the eyes, the retina propria, and visual cortex. It goes through this part of the LGN, of the thalamus called the LGN. There's another thalamic area, LP, that's sort of the equivalent of pulvinar in primates and in humans. We can also record in the hippocampus, because automatically, as you record between the cortex and the thalamus lies the hippocampus, area C1, C3. We can record the running speed, the pupil width, all of that data is available. So here you can see the differences between the advantages and disadvantages in this little cartoon movie of recording from optical physiology and electrophysiology. So in optical physiology, typically you record in a plane. So here, this is, a plane. Let's see, the field of view here is like 200 by 200, 250 by 250, or can be up to 400 by 400. You record all neurons within that plane that express this fluorescent marker. And the advantage of this is expressed only in a genetic identifiable a subpopulation of all cells. So let's say, it's all excitatory cells, or it's all inhibitory cells, or it's a subset of inhibitory cells, or it's a subset, let's say, in layer 5 of excitatory cells that project to the thalamus. So that's a great advantage. You know something about the identity of the neurons using optical recordings. The speed itself, it's slow. So the camera and the imaging is done at 30 hertz. So you don't pick up individual spikes. And in fact, and Saskia knows probably more than anyone else about this, because she's published several papers on this, trying to link the activity of spikes with the activity that you pick up using these 2 photon calcium imaging. It's a complex nonlinear relationship, but in general, it stands in for neural activity, the calcium signals that you pick up. The other advantage is you can come back, because you can see these cells here, you can come back the next day, or even, let's say, 10 sessions later, and still pick up the same neurons if you have appropriate software and for that. So you can track neurons over multiple sessions. Now, you can't do that with electrophysiology. Electrophysiology, you're essentially putting, a microphone, as it were, electrical microphone into the brain. It's very small, and it picks up neurons within its listening radius, typically within 50 or 70 micron of that electrode. You don't know their identity. Sometimes you can say whether they're thick spikes or thin spikes, and that tends to relate to whether they're excitatory or inhibitory. The advantage is you pick up individual spikes, and we know a lot about spikes and the genesis of them, and because spikes are ultimately what's transmitted to other neurons, and you can pick them up at very high temporal fidelity. So typically 20,000 or 30,000 hertz compared to 30 hertz. So it's 1,000 times higher bandwidth. And you can do it throughout the entire brain. So this little movie shows you, you know, you can record, so the cortex, here's hippocampus, C1, dental gyrus, C3, and there you pick up LGN, the medial geniculate, or you can even go lower. Here, in this cartoon, each circle is sort of is proportional, the diameter is proportional to the baseline firing rate of this neuron. So this tells you something about the advantages and disadvantages of these two different techniques. So here, for example, it's streaming. This is from one experiment, recording from these 6 different areas. And so, you know, if you do this for, you know, if you do this over 10 or 20 sessions, you get a lot of units. Units here just means neurons. You get a lot of neurons. You know, typically when you go back in literature and read some of the historic papers, they have 30, 40, 50 neurons. And now you can do things with 10,000, 20,000 neurons. That has the huge advantage that the statistical validity, it's much, much more easier to pick up with very high statistical validity, you know, relationships that before you just couldn't identify because you didn't have the numbers. Plus, of course, you can also pick up the dizzying variety of different neurons. All right, so first of all, we have to prove to ourselves and to our, you know, colleagues that these neurons are visual. And they do all the, I'm not going to go into this, they do all the standard things that you expect of neurons. Let me see, do I have a pointer here? No, I don't. So here, for example, on this lower histogram, again, it's something, you know, that you couldn't do before because people typically only report selected neurons. Here, the nice thing about a pipeline and doing this, you know, under standard operating procedures, whenever a neuron passes some criteria, you know, it gets recorded. Whenever it passes through, you know, the QC criteria, we record it and analyze it. And so then, unlike in typical studies where you move your electrode till you get to what's called a good neuron, and then you just spend your time with a good neuron. Here we record everything that we can pick up within the listening radius. So you get a great diversity of cells. Many of them don't behave according to the standard criteria of how neurons should behave. Let's say a simpler complex cells. But that's, you know, we pick up everything that's there. And so you can see here on the lower right in the histogram E, you know, so this is, for instance, a fraction of neurons that have actually visual receptor field. It may well be possible that not every neuron actually in visual cortex has a direct receptor field. In hippocampus, they have almost none, as you expect. So that's just as a control. So these are neurons. They have receptor fields, in other words, places in the visual field from which, if you put a stimulus in that region, the neuron can be driven. They have onsets to light flash, to dark flash. You know, by and large, these neurons behave as you would expect them to behave. Now, in an earlier paper, or in fact, in a series of papers that we published over the years in more than 2,000 mice, we've done very detailed analysis of the connectivity of the mouse brain using both wild-type animals as well as animals that express genes, or using a viral technology where we inject a virus into a part of the brain that expresses only in particular subtype of cells and carries a fluorescent tracer. So we can now, and that fluorescent tracer goes all the way along the axons, all the way to its presynaptic terminal in the region of the brain where it projects to. So we can get this whole brain connectivity we call this mesoscopic connectivity, unlike the data that we just released a few weeks ago, which is microscopic connectivity at the electron microscopic level. It's mesoscopic since you see the entire cell population, let's say in layer 5, that projects to thalamus, not just individual cells, but the entire population. So you don't get the single cell resolution, but you get the resolution of cell types. So on the rows, each row is an animal where we injected, you know, a tracer into a particular part, either of cortex or of the hippocampus or the hypothalamus or wherever. You know, you can see these different colors. And then we reconstruct the brain, the fluorescent signal proportional to the area where it projects to throughout the entire brain. On the left, it's it's the ipsilateral, the same side where the injection was, and on the right, it's on the contralateral. And so you can see, you not only do you get a gestalt, so for example, cortex has a high connection within cortex. Typically there are many more connections along the diagonal. Most areas, have massive interconnections, but also you can see the pattern of connections across the brain. So, and people this is sort of one of the standard tools that people use. This is the advantage of using an animal like the mouse, because we have all this wealth of data available, not just for any one particular area like V1 or some other sensory area or your other favorite areas, but for the entire brain. So we've done a deep dive on that several years ago, Julie Harris and Stefan Mihala. So these are people from the anatomy part and from the modeling theory part of our institute, of the Brain Institute. And we are using machine learning because we have access to the detailed patterns of connectivity. So, when axon goes, let's say, into another area, does it go only does it only make connections, make presynaptic terminals in the middle part, or does it also go to the upper part and the lower part of that area? That tells us whether it's an intermediate, whether it's a feed-forward connection, or it's a feed-backward connection, or it's so in this case, we classified connection as either feed-forward or feedback using machine learning, and then we arranged all the areas. So here we have 37 cortical areas and 24 thalamic areas on one side of the brain. We arranged them into hierarchy. We computed the optimal hierarchy. And so we assigned this. You can see on the bottom left, we have this axis, the x-axis, that assigns a hierarchy score. So a hierarchy score of 1 would mean it's one step in for instance, the Hubel-Wiesel hierarchy, simple cell. That would be a one-step hierarchy, going from LGN in one step to in a single unambiguous projection into visual cortex. And from V1, if you then project from simple cells, let's say, to complex cells, that would be a hierarchy of 2. So the mouse sort of has a flat hierarchy. Everything fits between -1 and +1 there. And this is, as I said, cortex and thalamic wide, because there are many, many parallel connections and there are many backward connections. So what we wanted to do with this first neural pixel survey data is to see how can we relate the anatomy, these anatomical hierarchies. So we see a clear anatomical hierarchy.
Can I ask a quick question?
Of course.
How do you interpret the fraction score? Like the 1 and 2, it's kind of easy to understand. But what about the 0.5 score? What does that mean?
The fact that they are also massive, that there are multiple steps in this hierarchy, as you can see here, right? There are multiple steps and there are multiple pathways, there are multiple parallel pathways. So it's a very complex system. It's not just a simple hierarchy. For us humans, it's very simple to think about a hierarchy. Like in a business or in an army, right? You always report to a single person, so you can see from the bottom to the top, there's 10 stages of command. But of course, in real life, there are also parallel pathways, there are informal arrangements, same thing in the brain, in particular in the small brain. This is, in a monkey, let alone a human one, it's expected to be, if you analyze all the data that no one has done, because no one has all the data, that you will probably get a deeper hierarchy, with 5, 6, 7, 8 levels. So it just means there are many bypasses, parallel pathways. An area sometimes can have lots of feed forward and feedback, but it may have a predominance of feed forward versus feedback. So then it becomes an intermediate area. It's a complex system. It's a very complex system, the brain. I like to call it the most complex piece of highly active matter in the universe.
All right, thank you.
Yeah, so you can see, for example, here on the top right, the blue one, we've just looked at the hierarchy. We've just sort of taken out the hierarchy for the visual system that we're now trying to relate with the neural pixel data. And you can see there are lots and lots of pathways. So if it were like a ladder, then it would be a clear hierarchy, right? A goes to B, goes to C, goes to D. But in reality, there are all these overlaps. So here again, same thing on the left. So what we try to do is, can we see evidence of this hierarchy in the functional data? So we have structural data, and now let's look at functional data. So in particular, if you look at the cross correlation, so in B, we have two neurons, let's say in V1 and LM, that we record simultaneously with these neural pixel, and we can see the cross correlation, right? So there's a peak in the cross correlogram. In this case, it's offset by 3 milliseconds, and the offset is positive. In other words, whenever the V1 neuron fires, 3 milliseconds later, the neuron in area LM, a higher order visual area, will fire. And so therefore, we interpret this, if it's within a particular temporal duration, if it's sharp enough, we interpret this as a connection from V1 to LM, right? So we can assign, depending on the sign of the offset, we can assign either it's positive, that means it goes from, or it's negative, it receives input back from that area. So here in C, we do that for all pairs of neurons between V1 and LM, and we see by and large all the peak offsets are positive, not always, because neurons are neurons and there's lots of variability in it, but on average, in this case, we have a 6 millisecond offset. And so we can compute the median cross-correlogram time lags in the cross-correlogram. Then these are shuffle corrected to account for slow drifts and shifts. And then what you see in D and E, we compute the anatomical hierarchy score difference between any of these 6 cortical areas and any of their other 6 cortical areas. Just a difference in hierarchy. So blue means they receive feedback and their feedback connections, and red means their feed-forward connections. And then in E, we do the same, but for the functional. We look at the time, the average, the mean peak offset. And blue indicates feedback, when the offset is negative, and red indicates feed-forward. And then, we simply, you can obviously see by eye, they seem to be overall very similar as one might expect. And here we have this nice correlation between structure, the anatomy, and function. So in other words, V1, on average is at the bottom, and areas, the medial area, posterior and anterior medial area at the top of this hierarchy. Now we can find lots of other correlates of this hierarchy. And this is the beauty of having so much data. You can find all these very quantitative lawful relationships that you could never pick up if you had 50 neurons. So for example, in the top row, A, B, and C, we look at the post-stimulus time histogram, when do neurons start firing in response. In this case, I think it's a full field flash. And here you can see the average post-stimulus time histograms of neurons in LGN and V1. And remember, LGN and LP are the 2 thalamic areas. The other 6 are the cortical areas. And you can clearly see the first neurons that fire on average always is LGN, as you would of course expect, followed by V1. So then we can compute the time to first spike. And this is the histogram of that over all neurons, 30,000 neurons. And then here you can compute the correlation between the average mean time to first spike in these different areas. And you see the colors throughout this paper are always the same. This light pink is LGN, the violet is, the violet purple is V1, and it goes up to, area PM, ending with PM, the 2 medial area, posterior and anterior medial. So it's a very nice linear relationship. You can look at the mean receptor field size as you would expect in a hierarchy. As you go to higher and higher areas, the receptor fields become larger. We also look at measures of phase. How much are the neurons driven at the dominant frequency? So let's say you have a drifting grating. If it's a simple cell, standard tubular visual simple cell, it should be perfectly well driven, modulated by the frequency of that grating. And so we compute what's called a modulation index. numbers close to 1 and take the log of that correspond to simple cells, and then numbers close to the log, the smaller they are, correspond to complex cells. And so you can see a systematic shift from being phase-driven early on in V1, where you have many neurons that have a log modulation index close to 1, to cells that are less and less driven by the dominant frequency. But as you can see, it's very diverse. So that's one challenge. If you look at many neurons, real neurons, many of them don't really behave according to standard models. A few ones always do very well, but if you look at all of them, they don't. At the bottom, we look at a prediction of C.J. Wang and colleagues, where we look at that the time constant of neurons, as it's driven by activity, in this case flashes, will systematically increase as you go along up the hierarchy. So here what we do, we look at the autocorrelation of the driven activity, falling flashes of light, and we fit exponential decay through the autocorrelation function and then get a time constant. So you can see that in J, in K, we do the population histogram, because we can do this for each of these thousands of neurons. And then on the right, we have the correlation, where we correlate the position of the hierarchy with this time scale, the autocorrelation time scale. And what you can see, it's a nice relationship that we do not see in the spontaneous activity. So if we do exactly the same thing, otherwise we fit an autocorrelation function, we compute the autocorrelation function of spontaneous activity, not driven activity, and fit an exponential through there, we get these time constants that are roughly 70 milliseconds that are. So the individual neurons, their autocorrelation on the order of 70 milliseconds doesn't depend on where they are in the position. But if you look at the autocorrelation of driven activity, that becomes slower as you ascend the hierarchy. There's recently a paper that does the same thing in intrinsic imaging in fMRI and finds the same relationship, only the time constants are roughly 10 times higher in the human brain. Very nice, very satisfactory. Then lastly, we do behavior. I think Saskia is going to talk more about it. Here we have an animal that's trained to detect changes of images. So you can see that these images that flash up, and occasionally it changes from one natural scene to a different one, like there. And then if the animal detects that and licks, it gets rewarded. So they're slightly thirsty, we water deprive them, and so they're highly motivated to do this, and they're getting very good at it. I mean, once you train them, they're very good at it. And so now the difference is, before it was all passive. The mouse is running and you flash up images and you have no idea whether the animal cares. I mean, why should it care? Because it doesn't get rewarded based on any images. Here, the animal has to care and does care, because it does its behavior and does it quite well. So you can look at the response. You can compare it. So you can look at images, how the cells respond to images. Particularly, you can look at images when there was a change in image, right? When you have the same image as part of a series, or the image is shown as the first image in the change, which the animal has to pick up, and then look at correct versus incorrect. So here on the right hand, in panel D, you can see the response, same image. So image identity is the same, but when it's not part of a change, so you get the lower response, or when it is part of a change. And when this is an active behavior, not surprisingly, when the animal detects it, the change signal, the signal becomes much bigger. The neuron responds more vigorously in the context of a behavior when the animal detects it, compared when it's not part of a change sequence. So we quantify that, call that a change index. The difference over the sum of those responses. And then Oh yeah, we also find the, oh yeah, so here we look at, on the bottom, we look at the hierarchy score and the change module, this change modulation index. And as you go up the hierarchy, this change index becomes larger. So that's important. What does that tell us? That tells us as you go from LGN to V1 up the hierarchy all the way to AM and PM, the signals are more and more modulated by the visual, by the behavior. And we compare the dotted line, the solid dotted line versus the gray out dotted line. There, the difference is it's active versus passive. So what we do, active is when the animal is engaged in a task and correctly detects it. Passive is when we've recorded that sequence now, so we show the animal the exact same sequence, and then you don't get that nice relationship, or it's much weaker. All right, so that's important. As you go up the hierarchy, the behavior, in the upper parts of the hierarchy, there's much more modulation by behavior and supposedly cognition, and in the lower hierarchy. Here on the right, this is, I think, my last slide here for this part. On the right, we correlate the anatomy, again, the hierarchy against the correlation of a decoder. So what we do, we use, random forest decoding of, 20 units that we do, 1,000 times. And then we see what's the correlation of that, of the, when you're looking at random neuron, at neurons in these different cortical areas versus what the animal actually did on that trial, right? So you compare on this one trial, I can decode the stimulus with 80% probability, but the animal wasn't able to decode it because it ran past the change detection, right? So I can do this. And here the interesting thing is, so you get effectively at LGN, there's no correlation between the decoder and the animal's behavior. Although if you just look at purely the decoder, it has, chances 50%, so it gets 68% and correct. So what this shows you, that just because, and it's a mistake a lot of people make, just because you can decode something, right? So today with machine learning, you can decode all sorts of things, but just because your network, your decoder can pick up some signal, whatever it is in one particular area, does not at all imply that the animal actually takes advantage of that information for its behavior. So in this case, although the animal could already, the information, some of the information is already present in the LGN, it's not related at all to the animal's behavior. So the animal doesn't use that information. That it only starts using information in the higher parts of the visual hierarchy. All right, so the last part, so over the last several years, we thought a lot about how can we make this fantastic tool, or the set of tools, this pipeline, available to the general community. And we call this open scope. And so we just received, so we did this, I'll show you one slide. Over the last 3 years, well, 2 years before the pandemic, already for select people who attended our workshop. But now it's open worldwide to anyone. You don't have to be even at an American university. Anywhere people can apply to this and to the open scope. And here the goal is to maximize the impact of our platform, to build and support an inclusive community, because now you don't have to be at MIT or Stanford or UCL, at a place that's rich enough to afford all these tools, but you can be anywhere. If you have an interesting idea and you have the means to, and the know-how to analyze it, you can write a proposal. It goes through double-blinded peer review, and then we collect the data. You only have to commit yourself to analyzing the data. And of course, it leads to this open, and we make all the data available to you, but after 1 year also to everyone else. So it leads to this, open in terms of, the reproducibility crisis. This is one way to overcome it, because the data is all there. It's high quality data, and anyone can look at it. If you don't trust the results, well, here's the original data. Just, do your own mining. So the entire pipeline is open to the community, from transgenic mice to surgery, everything I just told you about, and then either doing neural pixel experiments or doing multiplane 2-photon imaging. Right now, we are limited. In the first couple of years, we're limited to vision and visual cortex. I mean, that's the strength and the weakness of a pipeline is it's highly reproducible, but it's also much more difficult to change the pipeline. You know, we can't just sort of on a term of a dime say, okay, we don't want to record from visual cortex. Now we want to record from olfactory cortex. That would require retooling. We're getting there. We're setting up devices, so we should be able to do this brain-wide, but we are not quite there yet. And then all the data will be released. You know, it's all done through the cloud. So because this data is big, you know, we're talking about many gigabytes, even simple files here. So you can do all the cloud analysis, including machine learning with lots of tools for that. It's all in the cloud in standard NWB protocol. We do yearly application cycles. So we'll do 4 of them. We have done this already in the past, you know, just pre-pandemic in 2018, 2019, and there are a bunch of papers that have come out of that or that are still in progress. And we just opened up the request for proposal. So anyone, all of you could also apply to it. So there's a letter of intent. I think it's like 1 page or maybe 2 pages. The full proposals then is due in November. You can go to this URL. You can just Google OpenScope RFP. If you just Google OpenScope RFP and you get to it, and we have a workshop to explain. But the basic idea is, so you propose something, it then gets double-blinded review. So we don't know the identity. You shouldn't identify yourself in the proposal, just to, you know, to get to, because there'll be many more proposals than we can possibly run. We are funding for running 4 experiments a year for these 5 years. And then you have to commit to a data analysis plan, right? Because we don't want to go through all the pain and you know, the work of generating the data without being reasonably sure that, you know, you have some very precise idea of what is it you want to test. So that's out there. I think that's pretty awesome. The first of its kind, and hopefully will be more like our colleagues, the astronomer. All right, in summary. So I hope I told you a little bit about the brain observatory. Saskia will tell you more. That allows us to record under these highly standardized conditions in a high resolution 3-dimensional coordinate system and make all this data freely and openly available. We've now moved to this OpenScope environment. I told you about the hierarchy. And yeah, so we've just released a lot of data as part of this change detection paradigm, and including all the behavioral analysis. Most of the data has not been seen yet by human eyes, much of that. So there's a lot of stuff to do that we hope the external community will discover. And with that, this is part of the Allen Institute team. So it's a large group of scientists focusing on a variety of questions in neuroscience, but also in cell science and immunology. And of course, this was all started and set up and endowed by the generosity of one individual, Paul G. Allen, who passed away several years ago. And with that, I thank you for your attention.